Hi, everybody. Thank you very much for coming. My name is Nicholas Fisher. For those of you who don't know me, I'm a professor in the School of Marine and Atmospheric Sciences, and I'm also the director of CIDER, or the Consortium for Interdisciplinary Environmental Research, which is co-sponsoring this talk with the Provost. The Provost sends his regards, was unable to come today. He said his schedule has gone sideways. I don't know what that means, but he was not able to be here. But it is my delight to present to you no presentation is necessary, but for others who are meeting Paul Falkowski for the first time, I hope it will be an interesting experience for you, as it usually is for most people. Paul is a New York City boy originally, and went to City College, got his PhD back in the last century at the University of British Columbia in Vancouver, postdoc at Rhode Island, University of Rhode Island, in a very good oceanography program there, and then moved to the Brookhaven National Laboratory, about a half hour away from here, where he rose up in the ranks in what was then an oceanography department that was supported primarily through the Department of Energy. And Paul really built his reputation through some very landmark studies on a variety of things, but he was originally best known for his work in photosynthesis, marine photosynthesis, physiology and ecology of marine phytoplankton, single-celled plants that are in the ocean that account for about half the oxygen we breathe. His work has been widely recognized, and then, but when the Department of Energy said, you know, we're not interested in remaining in the oceans anymore, Rutgers University was wise to grab up Paul, and Paul has been a chaired professor at Rutgers for many years now. As I mentioned, he's been recognized through altogether too many awards. I think he should start shedding some of them, but he's a member of the American Academy of Arts and Sciences since 2002 and the U.S. National Academy of Sciences since 2007. He's also been an elected member to various learned societies such as AGU. He's won the Huntsman Award for Excellence in Marine Science, the G. Evelyn Hutchinson Award, through American Society of Limnology and Oceanography, the European Geosciences Union Vernadsky Medal, the ECI Prize from the Ecology Institute in Germany, and I believe a number of others, and probably some that are soon to be announced. He's published extensively in nitrogen and iron cycling in the oceans. He's produced some seminal studies in marine corals as well, which is somewhat seemingly different from algal physiology, actually not so different. And recently, in the last few years, he's been working on the evolution of life on Earth, and I believe that's going to be the subject of today's talk. And I first met Paul in about 30 years ago at Brookhaven National Lab when I was there for a couple of years, and there was something hauntingly familiar about Paul, and I couldn't place it. And I realized we discovered, actually, when we were talking, that we actually crossed paths over 60 years ago when we were both infants living born on the same street corner in Manhattan, 123rd Street and Amsterdam Avenue. So I knew there was something familiar about Paul, even when I saw him at Brookhaven. So my pleasure to introduce Paul, and I'm sure this is going to be an exciting talk, and it's my pleasure to introduce you.
Thank you very, very much, Nick. Thank you very much. And thank you all for coming. So the title of my talk is based really on a title of my book, which I published a couple of years ago. And I had been thinking about life's engines, if you will, probably for 20 years. And what I'm trying to get down to, what is an engine? I'm not talking in a metaphor. I'm talking about the actual protein machinery of life. And that's what I've been trying to understand, its evolution, its history, and how it came to transform the planet. So I'm going back to the beginning. These are amongst the oldest rocks on Earth. They're from Greenland. And this is the Ishua Formation. I'm standing at the top of the Ishua Formation. The Ishua Formation is about dated to 3.8 billion years. It's famous because at the bottom of this formation, there is the first sign of, quote, organic matter. It's really graphite. So it's pure carbon. And the isotopic ratio of that carbon is indicative that it was potentially put there by life. It's isotopically light. Contains less carbon-13 than carbonate. At the top of the formation, though, what you can see are these banded structures. This is not exactly banded iron, but it's similar. And you can see that there are areas that are obviously eroded, but are also oxidized in terms of iron. So that's rust, interspersed with black areas, which are laminae of reduced iron. And for those of you who aren't geologists, when you see striations like this is almost the classic example of a sedimentary deposit. So this deposit is representing, to first order, a marine system that existed when these rocks were formed 3.8 billion years ago. So we had liquid water on the surface. We know we actually had liquid water on the surface of this planet from about 4.2 billion. We're not exactly sure, believe it or not, it's something we don't tell the children in marine science. We don't know exactly where the water came from, amazingly enough. Now, before I get into water, I want to go through this very, very, very quickly because it's a very interesting diagram. This is the life work of Victor Goldschmidt. Goldschmidt was a geochemist who lived most of his life in Norway. He wasn't born in Norway. He was born in Switzerland. But he was a Jew who emigrated to Norway and spent, with a very short period, during the Second World War, he was taken by MI5 to London because he had been captured by the Gestapo and was to be deported to a concentration camp. And the MI5 recaptured him, took him to London. He lived in Europe, in England for several years, and then after the war he returned to Norway. And his passion was to determine the distribution of elements in our neck of the universe. And he didn't know at that time that the hydrogen and helium were primordial elements, were the original elements that were created from the Big Bang, so they're about 13.9 billion years old. And then we come to this lithium, beryllium, boron hole. This is an interesting problem in geochemistry. We know why that is now, but he didn't know then. And then we come to carbon, nitrogen, oxygen, and we walk around here, and phosphorus and sulfur are there. So hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur are the big 6 elements that make life up, with a sprinkling of a few other things. One of the other things are transition metals. So there's iron is the most abundant transition metal on the planet, and in the general cosmic area of this universe. And I'm just going to mention in passing, there are 2 elements that are missing because they do not have stable isotopes. It's not that they're not made. They exist, but they decay before you can measure them, basically. And then, of course, we have uranium and thorium and potassium-40. And those 3 elements are really critical, and they'll come into the story later, because potassium-40, uranium, and thorium are the major elements that drive the heat engine on our planet. So they are the radioactive decay elements that lead to tectonics. Now, the origin of life is really an invention of what we call non-equilibrium redox chemistry. So I won't describe what this is yet, but it involves 5 of the big 6 elements. So there's hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur. You can rearrange this and call that schnapps if you want to remember it. The only element that doesn't exchange electrons here is phosphorus. Now, redox chemistry is the exchange of electrons with or without protons. So we're sitting in the room now, and we're going to all go to an experiment to show you that you know what redox chemistry is. So life is electric, and that also is not a metaphor. Every cell in every one of our bodies, or every cell in every body, every living cell has an electrical field associated with it. We can measure that electrical field. So it's not just nerves, it's not just muscle cells, it's a microbe has an electrical field associated with it. And they use the electrical field to generate energy for growth by moving electrons from a substrate to a product. So therefore, substrates and products must be cycled. So that means all biological processes come in pairs. Every one of the cycles has something producing a substrate and something producing a product. And the product and substrate are coupled biologically. So the classic example we teach even in 4th grade is photosynthesis and respiration. And so let's take a look at photosynthesis before in the Archean Ocean, that 3.8 billion years ago. So what was the source of the electrons then? It was hydrogen gas. That was probably the earliest source. Iron 2, which is reduced iron. Hydrogen sulfide, that's a gas that smells like rotten eggs. And carbohydrates themselves. And you say, oh, Paul, where did carbohydrates come from? Well, believe it or not, chondritic meteorites contain an awful lot of reduced organic material. Amino acids, carbohydrates, and other things. So we actually import organic matter from space onto the surface of the planet in the early Earth's history. Today, the major source of electrons is water. We're a water planet. Now, what does that mean? So this is what a plant outside in Stony Brook is doing today. It's taking some water and carbon dioxide, and it's making a sugar. That's biochemical notation for sugar, and its waste product is oxygen. Now, you and I are in this room. We're breathing, and what are we doing? We're taking the stuff we ate for lunch or dinner last night or breakfast, and we're combining that carbohydrate or sugar with oxygen to make water and carbon dioxide. So we've just put hydrogen equivalents onto this gas, oxygen, and made water. So you're doing that with every breath you take. It doesn't hurt, right? You're deriving energy from a very, very complicated reaction, actually. Now, if you want to have oxygen on a planet, and this is going to be critical later, if you want to have oxygen on a planet, you can't take all of the sugar and allow it to be used by organisms like you and I or bacteria and oxidize it. You have to hide this in a lockbox on a back shelf somewhere in Earth and prevent organisms from oxidizing the sugar. That lockbox will be rocks. So it's not enough to invent a machine that can split water and make oxygen. You have to also have some process that takes the carbon that's reduced as a form of sugar, for example, and bury it in such a way that it can't be respired. Otherwise, the oxygen would not accumulate. So these reactions are not balanced on a planetary scale over geologic time. Now, when we look at Earth, it looks like there's a lot of water. We say, and every student knows this, 71% of Earth's surface is covered with water. But it's a very, very, very thin film. And if I were to take all the water and just put it into one big ball, that's the volume of water that the entire ocean is. So it's really rather fascinating. We have a planet that's closer to the sun, immediately next to us, Venus, which has 96 bars of carbon dioxide in its atmosphere. We're at 1 bar. So if we were to stand on Venus, regardless of the temperature, it'd be like standing at about 1,000 meters in the ocean. You'd be squashed. But it's super hot because the tectonic processes on Venus have resurfaced it, giving that CO2, and made it such that you would make solder into a liquid lead instantly. This is why we don't go to Venus very easily. The other planet to the other side of us is Mars, and we're very sure today that at one point it had liquid water. There's no liquid water left on Mars. So that water went off into space somewhere some time ago, and amazingly enough, for at least the last 3.8 billion years, we've had liquid water on the surface of this planet And it's the only body in our solar system that has liquid water on the surface today. So we know that there's water on Europa, we know there's water on Enceladus. These are moons of other planets, but they're not liquid water at the surface. Now, water becomes really important in my story because I'm going to get to the origins of photosynthesis, but this is what has inspired me for many, many years. This is obviously from Notebook B from Darwin, a very famous little doodle, and it's the original sketch of the tree of life. And Darwin didn't know, I mean, it's an amazing, amazing thought. Amazing thought. So what we teach in evolution is descent with variation followed by selection, giving rise to new species through time, in a nutshell. Now, Darwin had no concept of the time here. He didn't know what time was. And he dared, at the very first edition, on the first printing of the first edition of Origin of Species, which came out on November 24th, 1859. And why did it come out on November 24th, 1859? It was a stocking stuffer for Christmas. 1859, November 24th. All right. He dared to make a claim based on the erosion of Kent, of the cliffs of Kent, that the Earth was on order of 400 million years old. And boy, did he get flack. He got super flack. So, the second printing of the book, actually is about the third printing of the book, he actually removed this calculation. And he didn't want this to distract from the main theme of the book, which was the idea of evolution through natural selection. Now we have many, many other ways of reconstructing the tree of life, and this one is an old one based on ribosomal RNA. And what you can see is, yes, we have this clade over here, which we call bacteria. This is all microbial. This clade over here, which we call archaea. This is all microbial. Most of this clade here, which we call eukaryotes, or group superkingdoms. And here, for example, are animals. That's all animals, right there. We're very closely related to fungi, which I know is true looking at some of the students in my class. And there are land plants. So when we look at this group, what you can see, we can see diatoms here, they're in the ocean, dinoflagellates here, chrysophytes, cryptophytes. They're not plants. They just happen to have the same machinery for making a living. Photosynthesis. Now, these organisms were discovered, many of them, the bacteria down here, amazingly, and many eukaryotic cells, were discovered by Lewin Hooke. And I put up here an illustration of Lewin Hooke's microscope. So I have never seen an original Lewin Hooke microscope. I know that they exist, but there are none left in Delft, and his house is gone. I went very proudly as a National Academy member to the Royal Society. I'm allowed to stay in the Royal Society as a guest. And I went to the librarian who I know, and I said, you know, Rob, I hear you have a Lewin Hooke microscope. And he comes back half an hour later with a white box with white gloves. He opens it up very proudly, and he gives me a pair of gloves, and I take out this little thing about that big, made of silver. And he says, it's a reproduction. He said, well, Lewin Hooke gave us 4 microscopes when he was alive, but somehow they've walked away with members, and they didn't return. So I have never seen the original one, but this is what he did. Lewin Hooke was really, really an interesting character. He was a rug merchant and a textile merchant, actually, in Delft. And he lived around the corner from Vermeer, by the way. And he made lenses. And this lens he made, he would take a piece of glass, Venetian glass, very expensive glass. He'd pull a thread, he'd break it, the thread, and then put the thread of glass back into the heat, and it would make a ball. And then he would polish it. And he never, to his death, told anybody how he polished those lenses. And then he made himself these silver plates or copper plates, and he could position some sample here, and he would just look through this lens, and I'm sure he must have been myopic. But the lenses were so good, he could get magnification up to about 400x. That's amazing. So Hooke, who had developed a microscope a few years earlier, could magnify only 12 times. So with this, Lewin Hooke identified animalcules. And these are animalcules. I love this one. So these were the eukaryotes, the single-celled eukaryotes. And what were the animalcules? The animalcules were thought at the time to have been spontaneously generated. These came out of the ether, if you will, literally. And they had little heads, little stomachs, little sex organs, everything just an animal would have. And for a long time, that was thought to be the microbes. It wasn't until 1836 when Cohen in Germany, in Breslau, which is now part of Poland, really rediscovered and had a microscope built for him that was a very, very, very high resolution microscope and began the world of modern microscopy and microbiology. And one of his proteges was Koch, and then, of course, Pasteur, and so we come into the modern world. Now, what was not realized until the middle of the last century, and it wasn't because of microscopy, it was because of other tools. So there were three other tools that came into, well, one of the other tools that was very important, of course, was the electron microscope. But protein crystallization and ultrastructures that were determined by X-ray diffraction. So Brookhaven National Laboratory is very famous for this originally. The original was discovered by Bragg in the sun, and the baby Bragg was the youngest Nobel laureate ever. I think he got a Nobel Prize when he was 26. And they established at Cambridge University then the Center for Structural Biology, which today is one of the most powerful centers still in structures in biology. So Venki Ramakrishnan, who used to be at Brookhaven National Lab, has won the Nobel Prize 3 years ago when he started out at Brookhaven National Lab, and the Department of Energy says, you know, we're not interested in the structures of ribosomes. So he went and he took Max Perutz's chair at Cambridge and won a Nobel Prize. You know, sometimes I wonder. So what does this machine do? This is a very complicated machine. This is this machine that splits water. This is obviously a cartoon. I'm going to show it to you in structure land in a minute. So it has 4 manganese atoms in it. 4 manganese atoms are required to split the water. This structure of the manganese atoms is not found anywhere in natural minerals. There are 31 minerals that contain 4 manganese atoms in a cluster. None of them will split water. So nature invented essentially a mineral motif. The electrons are passed from water ultimately through this thing, cytochrome B6F, which we have in our mitochondria. So you have this type of machine as well. And then we go on to this machine, and this is the second machine which pumps with light the electron to a very, very, very electronegative place where it can actually make a hydrogen carrier, NADPH, which is what is used to fix the carbon dioxide. Now I'm going to say 2 things about this machine. We don't really know how it works precisely. There are 6 groups in the world. It took 25 years to get the first protein structures for these 2 machines. It was Horst Witt that did this in Germany, 25 years. Now we can get these structures almost within a day, almost, in a synchrotron. We don't know exactly how it works, and the reason that's critical is if we could mimic that machine with earth-abundant metals as a catalyst, we could split water. And if we could split water, we get hydrogen. And if we get hydrogen, the energy economy of the planet is totally changed, totally changed. So it's not for lack of trying. There are countries that invest a lot of money in this, most notably Japan. And there is a Nobel Prize out there, and it may go to Japan next year, or may go to Germany. It may go to somebody in the United States, Petra Fromm at Arizona State University. Now, this is what the structure looks like, actually, of the reaction center in a ribbon-type structure. And there, we can turn it on its side, and there's manganese 1, manganese 2, manganese 3, and manganese 4. So we know the structure of this at 1.4 angstroms resolution. It doesn't get much better than that. And yet we still don't know exactly how that machine works. Now, when I say machine, let me explain to you what I mean by a machine. And so we did these experiments in my lab years ago. There's an effect. When we put a charge through here, I'm going to take an electron from water, extract it here, and I put it up onto a protein little carrier inside of this box here. Now I've got a negative and a positive charge that are separated by less than 100 angstroms. The voltage potential within that box is 3,000 volts per cubic angstrom. It's enormous. What is the result of that? The result is that that protein starts to collapse when that charge is there, because you have a negative charge and a positive charge, so they want to attract each other. So the protein literally starts to collapse. Now, when it collapses, it emits a pressure wave, a sound wave. It's called the photoacoustic effect. It was discovered by Alexander Graham Bell. He didn't do it with this machine, but he did it for something else. So if I take and I shine laser pulses, 1, 2, 3, 4, 5 pulses, and I synchronize all those machines, so they all get a bolt of light, transfer electron, bolt of light, transfer electron, bolt of light, transfer electron, what happens is all those machines start clapping like that together. That's a machine. It's making a sound. Now I can put, believe it or not, a hearing aid in a vial next to this while I'm putting these pulses of laser on. And I can measure the dB, the decibels of sound wave that is coming out of these machines. And I can convert the dB precisely to the amount of energy that's conserved in the photosynthetic reaction. It turns out that that machine operates at 35% efficiency. This machine operates at 80% efficiency. Very, very, very high efficiencies. And they're found in only one clade of bacteria. That machine is found in cyanobacteria, blue-green algae, pond scum. No other photosynthetic producer in the prokaryotic world exists that produces oxygen. Once. And those guys were inherited as plastids, chloroplasts, in the plants outside, or in this case in a single-celled diatom, or in a dinoflagellate, or in a coccolithophore. So they spread. The same machine spread to different body plans. And you call these guys plants and those guys diatoms and so on. They all have the same basic operating system. And here they are. So I don't know if you can see this, but these are the cyanobacteria. And just look at the yellow. There's the plastids. And you can see they're all over the eukaryotic tree. But that's the only clade down here that has them. Now, those machines are copied and 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 copied in different body plans. And then they start, in this particular case, to transform a planet. So you can watch these machines. They invaded land, they're in the ocean, they're in lakes, they're in ice, they're everywhere. Anywhere there's water and a nutrient, you'll find something that's photosynthetic that's splitting water. And that winds up to be about 45% of the productivity is in the ocean, 55% is on land, and it works out to be about 100 petagrams, that's 10 to the 15th grams of carbon is fixed per annum by the ensemble of all the photosynthetic organisms on the planet. And some very small fraction of that organic matter in the ocean is sinking to the bottom and will become buried, and some of it on land will become buried. But let's just talk about the ocean for a minute, because land plants didn't start until very late. The earliest land plants are probably at the end of the Silurian, beginning of the Devonian, about 400 million years ago. Before that, everything really is oceanic. And in the oceans today, and Bob Aller is probably more aware of this than anybody in the room, shallow seas and continental shelves become the major repository of organic matter. And that's an interesting phenomenon. So all of this little flux, there's a little bit of flux, less than 1 1%, much less than 1%, is ever going to be buried as organic matter somewhere in marine sediments. But then we get to this potassium, uranium, thorium story and tectonics. And this is one of those great thought experiments that was done by the late Twosell Wilson, a geophysicist at University of Toronto. And what Wilson thought of is this. There are two major types of rock that we have on the planet, two major units. So one type of rock we call a craton, and we're standing on one, a little bit of one now. And the archetype rock for this, it's got silicate in it, is granite. So that's what makes up continents. Now, you only have to know two things in physics here. That something that's thick doesn't conduct heat as easily as something that's thin. And if you put a match underneath, and the Archimedes principle, something that's light floats. So believe it or not, in the world of rocks, granite floats. So it's got more freeboard, if you will. So that's why we're above the ocean. Now, the ocean is got beneath it a crust which contains very little silicate. It's called basalt. So let's put a candle under here that's radioactive heat, and we start breaking this craton up, and we start to get two cratons that are moving away from each other, and we fill in the gap here with more basalt with a mid-ocean ridge. And we continue on. This is where we are in this stage of the Wilson cycle right now. This is the Atlantic Ocean. It's what's called a passive margin. We don't have earthquakes commonly, okay? On our coast or on the coast of the eastern portion of the Atlantic near the European craton. All right. At some point, the idea here is that this bit of crust that is in the ocean will start to get further and further away from the mid-ocean ridge. It will start to cool, and it will start to subduct. This is an active margin. And when it subducts, it will start to uplift some of that organic matter with its rocks onto land. And that will take it out of the Wilson cycle. So what am I doing? I'm taking organic matter that will be here or here or here, and I'm going to put it up onto the craton. And there it is. This isn't imagined. There it is, black shale. This is an example of a very, very famous black shale formation, the Toarcian black shale in Spain, right there. So we're in about the fifth known Wilson cycle. Now, once we put that organic matter there, that organic matter isn't going anywhere very fast. Nobody's respiring it. We've just taken the candy out of and put it in this lockbox. Now we can make oxygen. Without this process, we wouldn't have oxygen on the planet. And the oxygen, we're pretty sure, rose in several bumps. So we know that the beginning of oxygenation of the planet is dated now from a very obscure method, and I'm not going to bother you with it. But it's only been discovered over the last 15 years or so when this exact timing was, and it's about 2.35 billion years ago. It's called, euphemistically, the Great Oxidation Event. And then we think oxygen was fairly quiet at a low level. So this number here, this is about somewhere in the neighborhood of 1 to 3% of the present atmospheric level. And it rose then at a second burst, and we timed that now at late Cryogenian around 700 million years ago. And there was one final burst of oxygen in the Carboniferous when trees started to grow. So let's just talk about this in this metaphor. So here's the first half of Earth's history. We had a stable ecosystem that was only anaerobic. No oxygen was being produced in mass quantities. And 2.35 billion years ago, the ball went from the first ball into the second ball. So ecologically, we had two steady states with a major perturbation that occurred 2.3 billion years ago. And once we made oxygen, you now had a gas that could be distributed because of atmospheric circulation and oceanic circulation across the planet that could be used to very, very, very efficiently oxidize organic matter. It's much more efficient now than the anaerobic oxidation of organic matter. Now, several years ago, one of my favorite people is Tom Fenshel. He's a great microbiologist. He's retired now. And my colleague Ed DeLong, who was then at MIT, is now at University of Hawaii. I said, guys, let's draw the wiring diagram of the planet. So some of you in the room take biochemistry. You're familiar with metabolic pathways, Keg plots, these kinds of things. This is a wiring diagram of all of the electronic circuits necessary on Earth. I left a few things out. But basically, this is the photosynthetic routes. This is the photosynthetic pathway. This is what's producing the organic matter. And these are the consuming pathways. Now, I know it looks like that. Actually, that is a much easier graph to understand than the one I just showed you before. But it's very simple. We're just moving electrons around. It's just like we're moving people around on the London subway. So if I'm putting an electron in, I'm reducing something. It doesn't want to be there because there are a lot of other electrons in the car. When we get to the next stop, electrons get off, and electrons get off, and ultimately all the electrons will get off on the train. And it'll start cycling around and around and around. And that's basically, if you think of yourself as an electron, you can understand that pathway. Now, it turns out that there are only 400 genes responsible for the entire electronic circuit. And we know the pathways. And now I'm going to try something with you to think about this in a simple way. So we all took some kind of, well, maybe you didn't. I mean, I started out building radios as about 8 or 9 years old. But think about this. You need a power supply. So what's the power supply for this electronic circuit? It's the sun. We need some wires. So you have an atmosphere and an ocean. And then you need some diodes and transistors. And those diodes and transistors we see in body forms, but they're really only the manifestation of the machine. So let's take a look at what the body forms contain within them. About 60%, a little more, about almost 70% of the body forms that move electrons around contain iron. It's the most abundant, as I showed you, transition metal. Then about 15% contain copper, and then there's sprinklings of manganese, a little bit of nickel, a little bit of molybdenum, and the rest is kind of trivial. So here's an iron-sulfur cluster. This is one of the most famous and simplest machines that moves electrons around on the planet. We have this machine. This is called ferredoxin. It's got 4 irons and 4 sulfurs, and it's bound to 4 cysteines. And the monomer only contains 28 amino acids. I can make this in my laboratory with a different alphabet of amino acids that's not found naturally in living organisms, using L and D amino acids. And I can make a stable ferredoxin with only 12 amino acids. We can elaborate on that theme. And this is the enzyme that takes nitrogen from the air and converts it to ammonia. It's the only machine that does this. It's only found in prokaryotic cells. It contains 38 irons and 38 sulfurs. And in some cases, and in this particular case, you can see it right there, it contains 1 molybdenum atom. So this is an elaboration of the earlier machine. Why is this an elaboration of the earlier machine? Because I can just add 1 more iron-sulfur cluster here, and I can convert this from just moving electrons to being something that reduces protons to hydrogen, making hydrogen gas. I can make this in a hydrogenase. Nitrogenase is also a hydrogenase. And then we have these things that contain, for example, copper. When you talk about copper, you're almost always talking about something that is moving electrons to or from oxygen. So we have copper in the last terminal electron acceptor in our bodies, or donor. It's cytochrome C oxidase. I'll show you that in a minute. And then there are other superoxide dismutases. Now, when I told you there are 400 genes, if you actually look at the structures, it's ridiculous. these collapsed to 35 simple motifs. Nature didn't make a lot of different proteins with different motifs. It made a lot of different proteins with the same motifs. This is a Lego set. There are only 35 motifs. So it shouldn't be too hard to reverse engineer one power supply, 2 wires, 35 motifs, move electrons. And that's exactly what we did here. So we're continuing on with this. We have another paper that will come out in a few weeks, but this paper was published in the Philosophical Transactions in 2013. And this was the first scientific journal that was developed in 1665, I believe, by Robert Hooke. And it wasn't done because he wanted to have a high citation index. It was established in 1665 because he wanted to show primacy of ideas, because Newton stole one of his ideas on gravity. It turned out to be a pretty bad steal. So Robert Hooke, when he found out that Newton wrote his idea up and was going to be famous for him, he established the Philosophical Transactions of the Royal Society, and to honor him, Newton took every portrait of Hooke down and burned it. So there is not a portrait today in the Royal Society of Robert Hooke. It's amazing. All right, anyway, let's go back here. So this is that iron-sulfur cluster that I showed you in ferredoxin. This is the base of the tree, and there's the iron hydrogenase. And then we can walk off and 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 walk off. And here's cytochrome C oxidase. So this is a phylogenetic tree, if you will, of protein evolution by structure. I don't care about the organism. Now, I'm going to conclude this talk. I don't know if Martin Schunan is here? No. Okay, so you have a very famous geochemist who actually is now out at Brookhaven National Lab, I guess, as an associate director for, I think, applied science, right? So Martin was picking up for years, actually it's an old concept, but it's one we don't think about in geology too much. The concept that light itself, I told you there's a power supply, can move electrons in minerals. This goes back, actually, the most famous person that did this before Martin was a guy named Graham Cairns Smith, who was a professor of chemistry and geology at University of Glasgow. So this is the atmospheric, top of the atmosphere. This is what it looks like at the top of the atmosphere. By the way, these lines, that's not noise. Those are Fraunhofer lines. Those are lines of elements on the corona of our star that are absorbing light as it is emitted from the star surface. So that's the top of the atmosphere. Now, you see this band gap here? So this is the top of the ocean. So all this UV light today is killed. It is absorbed before it gets to the surface of the ocean. Why? Because we made oxygen that made ozone. So one gas has blocked the UV light. In the Archean Ocean, there was no ozone. And you know what? One of the more interesting things is UV light actually penetrates through the ocean quite efficiently. So let me go and do an experiment. I'm going to make a mineral. In this case, we're making it artificially. This is siderite. This is iron carbonate. It's very stable under anaerobic conditions. This experiment is done only under anaerobic conditions, no oxygen. And all I'm doing is shining a UV light here. And I don't know if you can see it in this room, but this is, you can see this yellow area, this rusted out area. We've oxidized the iron in the siderite with UV light. And it turns out that this is actually, in chemical terms, if you want to know what this is, we've occupied an antibonding orbital at 4.8 electron volts, which corresponds to 278 nanometers of light, not very deep into the UV. Now, what is the product? If you're a chemist and you're oxidizing iron, the electron has to go somewhere. The electron went to hydrogen. Now, why is that important? Because I just showed you way back here that one of the earliest metabolisms is based on hydrogen. All right. Now I'm continuing on with this, and why am I continuing on with this? So if I can make a ferredoxin with 12 amino acids, and it's catalytic, that means it can do the reaction again and again and again and again. This reaction is not catalytic. This reaction is sacrificial. I oxidize the iron, that's it, done, finished. Unless some other geochemical process reduces it. How did we go from this type of reaction to a catalytic reaction? That, to me, is one of the most interesting aspects of the origin of life. That's what I work on now. Can I add 2 cysteines to this, or some other simple amino acid, and make a catalyst, which is photochemically active? And that's what we're trying to do. So this is another one. This is the formation. Here we're taking manganese carbonate, and we can oxidize it to manganese 2, from manganese 2 to manganese 4. And you know what we make here? This is formic acid. We're generating an organic acid. So I am trying these experiments with one of my graduate students to try to make an amino acid. I think we can do that. So at the end of the day, why do we care about all these things? Well, you know, the mantra of NASA, there are 2 major questions that NASA has on its plate, which I think are some of the most interesting questions that we can pose to ourselves. The first one is, where did we come from? And the second one is, are we alone? Now, if I can make a catalyst, does that mean that I can discover the origins of life? No. Why? Because there is not necessarily a unique solution to origin of life. There could have been many possible solutions. And it's impossible from us now to reconstruct the unique solution that led to the origins of those nanomachines. I'm trying, but it's very, very, very hard to say that was the unique solution. But the other thing we can do is we can see glows from other planets, and this is an instrument set that did not get past the decadal review last time. We're coming up to a new decadal review, and that will be ending in 1 year from now. So what was this idea? The idea was to float an interferometer at the Lagrangian 2 point, which is the gravitational equidistance between Earth and the Moon. It's about 22,000 miles out in space, and that's where the James Webb Telescope is going to be flying, by the way. So this is an interferometer, and it could look at the dark side of a planet. And from the glow of a planet, we can determine in the infrared gas compositions. So there are a lot of people that are trying to figure out if I have, say, methane, which will absorb in the infrared, and nitrous oxide together, I would argue pretty strongly that that's a sign of life on a planet. It's very hard to get those 2 gases in my head on a planet that are not, they're so far away from each other in thermodynamic equilibrium without life. Now, we're doing this to some extent now, and I don't know, I mean, there are people at Stony Brook and people all over the place that look at extrasolar planets. Last time I looked, and I'm probably off by a few hundred now, there were about 3,800 extrasolar planets that were known. Now, most of these are too close to their parent star to be within the habitable zone, because that's how we discover them, is when they pass in front of their parent planet, usually. It's one way that we look. But we can also do what's called the limb experiment. So as the planet goes in front of its star, we can look at a very, very, very thin haze of atmosphere and determine the gas composition that way. That's what's being done now. It's very difficult to do this experiment. It's very, very difficult because the number of photons that you get and the number of contaminating photons that you get is very difficult to control. But I am sure that in the next 10 or 15 years, or even earlier, we'll know whether we're, quote, alone. And that's a very, very, very exciting time. Because what gases will be there? How were they made? What's the planet like? So that's really what Life's Engines is about to me. It's understanding how life changed the planet. It made engines, it evolved engines, that evolved other engines, that evolved other engines, but not very many. and they ultimately move electrons around on the surface of the planet in a gigantic circuit diagram. So, in the first 2.5 billion years of Earth's history, I call this the research and development phase, which is when a core set of metabolic machines evolved. This is like Turing, discovering how to compute. Okay? So all these key metabolic processes were developed in prokaryotes, very simple Turing-type machines, and there were only about 400 core elements. And then they were incorporated into other body plans. So that's when you have Steve Jobs and Gates and Dell and all these companies that come in and say, oh, that's this, and they're all using Turing-type machinery. I don't care what, you have a very sophisticated machine, but it's basically a Turing machine. So the basic problem here is it's all driven by light. And if I go and read books on the origins of life, most of my colleagues will say, no, it started off in a certain type of deep sea hydrothermal vent. Well, at some point we have to get to light. So I'd rather just get to light at the beginning because I know those reactions happened. They have to have happened. And if I go very back to my very first slide, you remember that oxidized iron at 3.8 billion years in Greenland? It turns out some of that iron is primordial. It's oxidized primordially. So I'm sure these reactions happened very early in Earth's history and were incorporated from light driving electrons non-catalytically in minerals to ultimately light driving life catalytically in proteins that contain minerals. So thank you very much.